C1 neurons drive chronic anxiety. Anxiety disorders currently affect more than 300 million people globally. While clinicians have mapped several broad brain regions linked to fear and stress, understanding exactly how these regions communicate to sustain chronic anxiety has remained a significant challenge. Because current medications often impact the entire brain and body, they frequently produce off-target side effects that discourage long-term use. A new study published in the journal Neuron provides a much more precise target. Scientists at St. Jude Children's Research Hospital have identified a highly specific group of epinephrine-producing cells, known as C1 neurons, as major modulators of fear and anxiety. The research team focused on a brain region called the rostral ventrolateral medulla, or RVLM. The RVLM is a deeply interconnected structure primarily known for controlling basic autonomic survival functions, like breathing and cardiac regulation. Because it handles such foundational operations, scientists traditionally assumed it did not house the neural machinery required to govern complex emotional behaviors like chronic anxiety. To test this, researchers developed a precision targeting system to isolate and interrogate specific subpopulations of cells within this region. They discovered that C1 neurons act quite differently from the cells immediately surrounding them. When a threat occurs, C1 neurons sound the alarm. They fire, subsequently exciting a downstream region called the periaqueductal gray matter, or PAG, which regulates physiological and behavioral responses to stress. Under normal conditions, this circuit is temporary. The neurons fire during a stressful event and turn off once the danger passes. However, the researchers found that strong or prolonged activation of these C1 neurons acts like a stuck switch. It forces the circuit to stay active, inducing an immediate anxiety response that can persist in mice for up to a full week later. Critically, the team also tested what happens when you block this circuit. When researchers inhibited the C1 neurons during a period of heightened stress, the mice were significantly less affected by subsequent stressful events. The inhibition effectively broke the cycle of prolonged anxiety. What makes this discovery particularly promising for future therapies is that blocking these neurons did not change the animal's immediate, necessary behavioral responses to acute stress, nor did it alter their baseline behavior. By pinpointing C1 neurons, this research opens the door to highly targeted treatments that can quiet long-lasting, pathological anxiety at its source, without broadly disrupting the rest of the brain and body. What do you think about this research? Drop us a comment below. Don't forget to like and subscribe for more neuroscience news research.